Hey everyone, today we've got Dr. Stuart Hameroff. Stuart is a professor emeritus, that's essentially the Yoda of the department, in the departments of anesthesiology and psychology, and is the director of the Center for Consciousness Studies at the University of Arizona. Today we'll talk about the Penrose-Hameroff theory, ORC-OR, or Orchestrated Objective Reduction, which describes consciousness as the intersection of biology with quantum physics. Then, how does this apply to aging, Alzheimer's disease, and even things like life after death? And I've got a whole bunch of other questions. So with that, first, welcome, Stuart.
Thank you, Michael. Nice to be here.
All right, so I think before jumping into the aging related stuff, I think it's important to go over ORC-OR and what it is, and microtubules and quantum effects, and how that links with consciousness.
Right. Well, most people would say the brain is a computer of simple neurons, a complex computer of simple neurons that interact by synapses. And these complex interactions, synapses, give rise to emergent properties, which include consciousness happening at a high level of complexity. I think that's bullshit, because if you look at a single cell like a paramecium, it's not an on-off state. It's a Well, I hate use the word complex because it doesn't, but it's its own thing. It swims around, finds food, finds mate, has sex, can learn, escapes from a capillary tube faster each time. Why is it doing all those things? And how does it do all those things? So it uses its microtubules, structures inside all cells, including our neurons, that are protein polymers of a protein called tubulin. So they're hollow cylinders of a lattice. So imagine like an ear of corn, and you hollow out the inside. Now you got the kernels making a lattice. And imagine they could talk to or communicate with each other like little switches, like little synapses. That's what microtubules do, except they do it much, much faster than neurons. They do it actually multiple frequencies, from hertz to kilohertz to megahertz to gigahertz to terahertz. So down into the optical range, they're actually interacting with photons. And they're in all of our animal and plant cells, including our brains, our neurons, where they're in particularly interesting arrangements. And they process information at the single cell level. So why wouldn't they be processing information in our brains, particularly when we don't have a better idea for where memory is stored? They store memory. They perform cognition. And in Alzheimer's dementia, microtubules fall apart. Alzheimer's is focused on these stupid amyloid plaques, which are ugly, but outside the neuron, and may or may not have anything to do with the cognitive dysfunction. There's some correlation between them and what are called neurofibrillary tangles inside the neuron, and those do correlate with cognitive dysfunction. And those are clumps of a microtubule-associated protein called tau that stabilizes the microtubules. And for whatever reasons, as we get older, the tau falls off, or the microtubules destabilize, and then the tau falls off. So you get clumps of tau, the microtubules disassemble, the neuron shrinks, you lose synapses, you lose memory, you lose cognition, and your whole brain shrinks. You've got Alzheimer's. And they're spending billions and probably trillions of dollars on these stupid amyloid plaques. Somebody's making a lot of money on drugs that don't work and are toxic, and it's the stupidest thing I've ever heard. You could treat the microtubules, and I've been trying to do this study at our institution, but I couldn't get any cooperation. By resonating microtubules with something very simple and inexpensive and non-invasive called ultrasound, which is used for imaging on newborns or fetuses in the uterus and newborns through their fontanelle to look for bleeds, and has been used in medicine for 100 years safely. At low intensities, it's painless. If you put it into your brain, you actually get a nice buzz and feel good for a while. And we did a pilot study where it improved symptoms in Alzheimer's. And I couldn't get any cooperation, and I think that should be done. You can do this with a low intensity handheld ultrasound device for about $150. And my dream, I'm getting kind of old now, but is to go around to nursing homes and go to every room and try it on somebody and see if they'll wake up. Kind of like that old movie about Parkinson's. I forget what they called it, Awakenings, with Robin Williams. But this would be, if you could repeat it, it would be permanent, or could be. And I think it's worth trying. So that's my main thing about longevity.
Yeah. So wait, I wanted to get into the Alzheimer's story later on, but I wanted to go through how quantum effects are happening in the microtubules, and how that potentially is the interaction between the quantum world and then what we perceive as the relativistic world.
Right. Well, so I got interested in microtubules when I was in medical school in the early 70s in a cancer lab studying mitosis, how cells divide and the mitotic spindles, which are made of microtubules, pull apart the chromosomes. And everybody else in the lab got interested in the chromosomes. It was the dawn of the genetic revolution and genetic engineering and so forth. They all probably became billionaires going into genetic, all that stuff. I found that really boring. And I was interested in how the microtubules knew where to go and what to do, that they had some kind of intelligence or consciousness. And I had been interested in consciousness since my undergrad days and was in medical school, was always oriented towards the brain, neurology, neurosurgery, psychiatry. But none of those lifestyles grabbed me. So I did some research and really got intrigued by microtubules. And to make a long story short, I did an internship and during which I was going to figure out what I wanted to do. And I met the chairman of the anesthesia department at the University of Arizona, Burnell Brown, who was, I told him about my interest in microtubules and consciousness. And he said, well, if you want to figure out consciousness, figure out how anesthesia works, because we don't know. And number two, here's a paper by a friend of mine that shows that anesthesia depolymerizes microtubules. I said, well, that's cool. And he said, and it's a lot of fun. You'll make good money. So I said, okay. So that was 49 years ago. I just retired last month from anesthesia after almost 50 years. And I really enjoyed every day of it. It was really, it was grueling. It was intense. It was sometimes exhausting, but it was fun. And the camaraderie with my friends and my colleagues and so forth. But anyway, I moved on and I'm now doing purely research in astrobiology, where we're looking for the origin of life and consciousness in the early solar system. But getting back to your question, so I was going, so I got the idea that microtubules were processing information as little computers. Like every of the kernels in the corn could be like a bit switching in megahertz. So a lot of information. In fact, the singularity people were saying, well, the brain has 10 to the 11th neurons switching with 1,000 synapses at 100 hertz. That's 10 to the 16th operations per second. And ironically, there's about 1 billion tubulins per neuron switching at 10 megahertz, which gives you the same 10 to the 16th operations per neuron per second. So I was going around to neural net and AI meetings being a real pain in their ass saying, wait a second, your goalpost is like 11 orders of magnitude further downstream. You're not going to be there in 5 years or whatever and even if you did, anyway, one day somebody said that to me. They said, okay, let's say you're right. We did have all this information processing. How would that explain consciousness? How do microtubule computations produce consciousness? And I was kind of stunned and realized that was a good point. I didn't know. And fortunately, he suggested I read a book by Roger Penrose called The Emperor's New Mind, which argued for a quantum effect. And he argued through Gödel's theorem that in Gödel's theorem, a mathematical theorem can't prove itself. You have to be outside the theorem. You need a conscious mathematician. So he said to understand something, to know it's true, you have to be outside. You have to have something outside the system. And that has to be quantum physics because it's the only thing outside of classical physics. And then he tied it to a collapse of the wave function in space-time geometry, bringing in general relativity. By then I was pretty blown away and I'm thinking, this guy is the smartest guy I've ever read. I mean, he is so all over the place, but so logical, so clear. And his artwork and cartoons explain everything so beautifully. I said, I really don't know, but I got a hunch he's right. And but he doesn't have a good quantum computer in the brain. For quantum, you need something that's going to be in quantum superposition of two or more states at the same time. It's one of the mysteries of consciousness. Of quantum mechanics. How can that happen? And he said that happens because he brought in general relativity. And he said, so, Einstein had this thing about space-time being curved. So if there was a star behind the sun, the starlight would be curved by the sun and we'd see it during an eclipse, even though it was behind the sun. And or it can only do it during an eclipse. And Eddington did the experiment and proved him right, showing space-time curvature. Eddington got his Nobel Prize and celebrated with Einstein, who already had his. But Roger applied this curvature to tiny particles, quantum particles, so that if a particle was here, it'd be a tiny curvature here. And if the same particle was here, it'd be a tiny curvature in the opposite direction. So you could, and he used these drawings of two-dimensional space-time sheets with little curvatures that started to separate when they were in superposition. And And then you can imagine that if those separations continued, you'd get many worlds. You get multiple worlds. So it happened again and again. And that's one of the explanations for superposition, that there's no collapse, there's no reduction. They just keep separating and bifurcating, and we have an infinite number of overlapping universes, which seems kind of wasteful and silly, but it's the most popular idea because it doesn't violate standard quantum mechanics. It doesn't really make any sense, but it doesn't seem to bother physicists. The math works. But Roger said, no, the separations are unstable and will collapse to one or the other at a time t. And when that happens, that causes consciousness. So let me back up, because the other possibility is when you had a superposition, that consciousness out there causes the collapse. The conscious observer looks at these separated space-time curvatures, and instantly, somehow, it goes to this one or that one. So consciousness out here causes collapse, putting consciousness outside the universe or outside of science. Roger said, no, the separations are unstable. After time t will collapse to one or the other and produce a moment of consciousness. So it was, and I think still, the only suggestion, possibility for a scientific mechanism for consciousness. Otherwise, it's all hand-waving emergence. And I literally hand-waving. I did, when I was in the 80s, I spent a lot of time with a good friend who was an emergence nonlinear dynamics person, Alwyn Scott. He wrote a great book about consciousness and emergence called Stairway to the Mind, kind of like the Led Zeppelin song. In fact, I gave him the title from the Led Zeppelin song. So Stairway to the Mind, and so you have layers and layers, and then you get to consciousness. Okay, that's fine, but you need bottom layers. Now they're just talking about emergence up here without any bottom layers. So I think it's all bullshit when they say that, unless they have a bottom layer, which has to be the microtubules. So anyway, I read Roger's book. I wrote him a letter and said, you need a quantum computer in the brain that can control neurons, and I think it's microtubules. And I sent him a paper. I told him about a book I'd written. And by the way, I was going to be in England for another conference. I'd be happy to come to Oxford. And he said, please do. So I think it was roughly October of '92. I met him in his office in Oxford. And he was very pleasant, very polite, very charming. And he told me he was going to a meeting in Cambridge with philosophers of mind, Dan Dennett and Pat Churchland, both of whom had gotten wind of his quantum idea and didn't like it at all. So he was happy to have something biological to at least mention. Or I thought, well, he didn't say that, but anyway, so I did most of the talking. I went through a book I'd written and showed him all the pictures. And his first question was, these are not simulations. These are real biology. I said, oh, yeah. And at the end of the several hours where I did almost all the talking, he thanked me very much. He said, thank you very much. And we shook hands and said goodbye. And I thought, wow, that was cool. I got to meet Roger Penrose. And little did I know that was just the beginning. So 2 weeks later, I was back in England and I was having dinner with a friend, and he said, I went to this conference in Cambridge and Roger Penrose was talking about you and your stupid microtubules. And that was a real thrill. And shortly thereafter, I got invited to a meeting in Sweden, north of the Arctic Circle, just for scientists, no audience, including Roger. And we got to know each other, played a lot of ping pong, went hiking in the midnight sun, and went skiing, actually, in the midnight sun, and became friends. And I invited him to the first Tucson conference in '94, and bribed him and his wife, Vanessa, with a trip to the Grand Canyon afterwards, which they gratefully accepted. And we hooked up. We met in Denmark a few months later and started working on our theory. So that's how it got started. We were working on a manuscript trying to quantify microtubules and ORCOR and collapse the wave function. It was going very slow, but then we got attacked in print by Pat Churchland, who wrote a very obnoxious, sarcastic paper attacking us. And the journal said we could respond in the next issue if we had a manuscript in 2 weeks. Well, we had been working for a year without a manuscript. Roger's wife told me I have to be very patient working with him because he's very meticulous. Everything has to be precise, and there's a lot of things going. But this annoyed him to the point where we put it together and we answered within 2 weeks. That was our first paper. And then the full theory came out a year later. So he was ridiculed by everybody immediately. The brain's too warm, wet, and noisy, blah, blah, blah. You have to be in absolute zero temperature, blah, blah, blah. And my first reaction was, so was Rogers. Well, the biology's had billions of years of evolution to figure this out. And the main way that we figured it out is how anesthesia works. Because everybody says, okay, the brain's too warm, wet, and noisy. Okay, wet. So our bodies are 70% water. Our brains are 70% water. Cells are 70% water. But the other 30% includes a lot of other solubility compartments. As an anesthesiologist, I give a drug, and where's it going to go in the body? So most drugs you give intravenous or you take pills and they get in the bloodstream. They're charged, so they're very soluble in blood or water, and they go to receptors on receptors on membrane surfaces. Anesthetics are like oil. Oil and water don't mix. They don't dissolve very well in water, very, very poorly, or in blood. So when you breathe, they do get in the blood and then quickly leave the blood to find something fat-like, something oil-like. That includes membranes and includes the interiors of proteins, which have aromatic amino acids like tryptophan, phenylalanine, and tyrosine, which all have quantum optical effects, photoelectric effects, where you hit it with an ultraviolet photon, you get an excited state, it's a quantum state, tools are on, and then it drops, fluoresces. And we've done experiments now showing this happens in microtubules and that anesthesia dampens this, but I'm jumping ahead. So, and I thought anesthesia solved the problem of too warm, wet, and noisy, because inside the nonpolar regions of proteins where these aromatic rings are, it's oil-like. It's not wet at all. In fact, it excludes water. They're hydrophobic. And in microtubules, these nonpolar aromatic pathways of, it's called pi electron resonance, the pi orbital of the aromatic ring, is continuous from along the tubulin and then from tubulin to tubulin. So you get these pi aromatic pathways that go the length of a microtubule, which could be meters in your sciatic nerve, or shorter in dendrites, but mesoscopic, if not macroscopic. And that solved the problem if they were oscillating coherently like a laser, which it turns out they are. And this was discovered later by our friend Anirban Bandyopadhyay, working at the National Institute of Material Sciences in Japan, doing really the most cutting edge stuff in the world. And he started looking at quantum oscillations in microtubules and tubulin and found these coherent oscillations that repeat every 3 orders of magnitude in microtubules. So people look at the brain and measure EEG, and we get these squiggly lines, and they're supposed to tell us something, and they're between 0 to 40 hertz, or maybe 100 hertz, cycles per second. And but if you look deeper with different methods, you see the same sort of patterns, not just in hertz, but in kilohertz, 1,000 hertz, megahertz, 1,000,000 hertz, gigahertz, 1,000,000,000, terahertz, 1,000,000,000,000, and even faster, every 3 orders of magnitude. And Anirban calls this, the microtubules act as time crystals. Time crystals were described by, I think, Frank Wilczek in physics, but nobody had a clue that they could happen in biology. And Anirban discovered that microtubules are time crystals. And he recently just published his 3rd paper on measuring megahertz and gigahertz from the scalp, buried in the EEG. So the EEG is well known. He calls it the DDG, dodecanography, for 12 orders of frequency inside the brain. So next year will be the 150th anniversary of EEG, and I think it's ripe for a revolution. And I think that this will keep going. We're going to be doing, involved with studies that Anirban's organizing in India in a hospital over the summer, where they're going to measure this as patients go under anesthesia and see if the megahertz goes away. And so I think this is going to change everything. And if it's not suppressed, I mean, our opponents have been actively suppressing, discrediting, saying we're crazy and blah, blah, blah. And I'm kind of getting fed up with it. And now that I'm retired, I don't have much to lose. So I'm fighting back. I know this sounds funny, on Twitter and a lot of other ways, but it's going to be interesting next couple of years. I think the mainstream in the neuroscience of consciousness, there's two groups, or 2 conferences. Our group, the Science of Consciousness, which is kind of broad spectrum. We include quantum, we include parapsychology if they have evidence, psychedelics, spirituality. near-death out-of-body experiences if they're well presented, but also rigorous brain science. The other group, ASSC, only considers what I call cartoon neurons. And this goes back to Christoph Koch at our first conference in 1994. He had been working with Francis Crick, whose idea was the brain's a complex computer of simple neurons. And he showed a picture of quite literally an empty neuron, just the membrane, a cartoon neuron. And I said, Christoph, why don't you look inside for the microtubules? There's all this other stuff going on. And he turned to me and he said, Stu, the persona of my favorite cowboy Western actor, Clint Eastwood, I'm going to answer, a man's got to know his limitations. And that's apparently a line from Magnum Force. And he was being funny and cute, and it worked because it kind of caught me off guard. I was back on my heels. I didn't follow up with like, how can you even believe this cartoon neuron thing? And it was Crick's legacy, and the cartoon neuron has lasted 30 years and led ASSC into a pitiful situation, I think, where they're selling out to AI because AI, I mean, these cartoon neurons are no different from AI. So if the cartoon neuron theories are right, AI has to be conscious inevitably. And that's what they're saying. So they're having these debates now, and they're concluding, well, compared, if these current models, global neuronal workspace, IIT, higher order thought, PC, all of them, they're based on cartoon neurons. If they're right, then AI has to be conscious. Therefore, we've surrendered. So go ahead, AI. So AI, being smart and wanting to control everything, I think, and particularly the narrative that AI can be conscious, because that would make it easier for AI to control things like the government and religions and things like that. So they're pushing this narrative, and the cartoon neuron people are playing right into it. They've just thrown the brain under the bus. I mean, it's all just a computer. There's no biology. It's kind of sickening, actually. Okay.
So no. That was all great. You checked off a whole bunch of stuff that I wanted to go through anyway. So
good.
So I too am in academia, and I see this often where, at every level, you've got a normal distribution of intellect and performance that goes with that. And sometimes you'll have scientists who you would expect that are should be the most open-minded, right? Data-driven and open-minded to what that could infer, right? But yet they're so regimented in their ways that they refuse to look at, outside ideas, even when they're well supported, like yours and Rogers, right? So then they hold on to those ideas, like they marry them as tightly as they can and hold on to them for the next 50 years. Now, anyway, without looking at things with an open mind. So it sounds like with the cartoon neurons and not considering microtubules and this whole equation, it seems like a lot of science is like that. And as you mentioned, you're literally fighting back. I see you on X, and I think it's great. I mean, I think more scientists should do this, and you do it in such a well-respected, respectful way.
Usually. I lose my temper sometimes.
Well, I mean, there are so many people coming at you sideways too, with all kinds of like ill intentions, and you handle it beautifully. And I've always tried to model myself in that same way, not knowing that you're doing that too. But look, you know how it is. Like, to break a paradigm, you've got to break some eggs, and sometimes it may take generations for the field to catch up to where you guys are, right? It's funny because when you read Roger stuff, I wasn't familiar with your work at all. I think I saw you on like Tim Ventura and Kurt Mungo, and you opened my mind. And I'm not an astrophysicist. My realm is physiology and biochemistry. So I wasn't even thinking about, quantum physics as it applies to consciousness. So anyway, all right. So question, right? So we know that there are quantum effects going on in microtubules, but then how does that link with the collapse of the wave function to, I guess, create consciousness in the brain? Is that what's happening?
Yeah. So consciousness, qualia, phenomenal experience has to come from somewhere. Darwin wrote a lot about consciousness, but he didn't include it in evolution because he didn't have a mechanism. It wasn't an enzyme. It wasn't something that could do anything it could see. So we haven't really had a scientific explanation for it. And accordingly, like in the 20th century or 19th century, the 20th century, it was deemed taboo in psychology. They wanted science. They wanted data. You can't measure consciousness. So behaviorists took over for most of the 20th century, and consciousness was thrown out. It was banned. And then in the late, like in the 1980s, it kind of come back due to books by Francis Crick, Gerald Edelman, and Roger Penrose, and even Dan Dennett, who wrote a philosopher. But the first three guys all won Nobel Prizes. And I think Crick was completely wrong about his astonishing hypothesis that the brain is nothing more than a pack of neurons computing. Kind of weird. The guy who figured out DNA has such a simplistic view of consciousness. But anyway, that was it. And he latched on to Christoph, who was an up-and-coming neuroscientist. And they became a team. And about the time that just before, actually, Roger and I became a team. Actually, Pat Churchland invited me to UC San Diego to, I thought, give a talk. And when I showed up, there was no talk. It was sitting around a table in her office with Francis Crick, Christoph Koch, and her grilling me about microtubules for 2 hours. And Crick fancied himself as a biologist, and so did Christoph. And I was kind of, I was prepared to give a lecture. I had slides and everything, and they're just like asking me all these questions about microtubules, see what I knew, I guess. And at the end, after 2 hours, Pat says, you bore up rather well. So I'll take that as a compliment, but she's been on my ass ever since, nonetheless.
Yeah, like pompous, right? What a pompous jackass and when he came at you with that line from that movie too, it's almost like he's coming from this intellectual privilege where he thinks that you're just not quote unquote good enough to be in his realm, which is seemingly ridiculous at this point, right? So
well, he does get a lot of adulation, or he has, so maybe that's what happens.
Right. I think history.
But he's been wrong. He's been wrong on everything. Okay.
Yeah, exactly.
He was wrong about gamma synchrony. He and Crick came out and said 40 hertz is the neurocorrelative consciousness, believing that it was due to spikes. So as you know, neurons have integrate and fire. And Christoph's always maintained that the firings are the bits, which gives you the computer. I've always thought the consciousness has to be in the dendrites. You integrate or orchestrate, you have a collapse, you have a conscious moment, and that triggers the spike to do what you're going to do about it, to speak or to move or do it, So the spikes are just carrying out, or like messengers. So that's what I've thought. So, okay, so he's wrong about, so he thought that gamma synchrony was due to spikes, so he was for it. Then he finds out that gamma synchrony is due to dendritic synchrony, so he was against it. So in 1990, Crick and Koch were for gamma synchrony. 1995, they came out against it because it wasn't spikes. So spikes were the thing. In his books, he always says, how else can you mediate without spikes? Well, microtubules, that's how, and dendrites. So, and then of course, IIT, he latched on to IIT, which has flamed out. I'm glad to say 125 neuroscientists said it was pseudoscience. Actually, their reasons, they're the cartoon neuron people who claimed that IIT was pseudoscience because it didn't even have neurons, not even cartoon neurons. It's just information. There's no biology in IIT whatsoever. So I think that needs to be put to sleep one way or the other. And recurrent processing in predictive coding is actually, I think, probably right, but it happens at multiple levels, including at the level of microtubules. So it's not really a mechanism of consciousness, it's a mechanism of biological information processing at all levels. And there's been a lot of really great work in the neuroscience of consciousness in the last year from Earl Miller's group at MIT, finding these EEG feedback waves going from the cortex to the back that seems to correlate with consciousness, and it meets incoming. And if you put an oddball in the incoming, the oddball gets selected. If everything's as expected, as predicted, nothing changes. So I think predictive coding is probably right, but it happens at a much deeper level. The other theories are all cartoon neuron theories or just pure information theories. So we've done, we made some predictions that we could demonstrate quantum effects in microtubules at ambient temperature and that they would be inhibited by anesthesia. So we were part of the Templeton World Charity Foundation adversarial collaboration program that you may have heard about, where I guess it was 5-6 years. They identified five major theories. and they wanted us to do adversarial collaboration, take two theories against each other, pick out an experiment that would refute one and prove the other one. Well, all the other theories are all level of neurons. And so it wasn't a very good fit. We tried to come up with something with IIT, and we had Christoph and Julio come to Tucson. But they wanted the experiment we eventually did of a fluorescence of a microtubule on an optical bench with canceling out any vibrations with lasers perfectly aligned. We eventually did this, showed quantum effects, showed they went away with anesthesia. They wanted us to do that in the brain of an awake monkey and then put the whole monkey to sleep. I'm going, no, I'm not going to torture a monkey, and it's going to be impossible to do. And no. So, and they said, well, okay, too bad. So they went on to spend $5 million in a inconclusive study with Global Neuronal Workspace, and Templeton gave us $100,000 to do one experiment, which we did, and it worked beautifully. We showed quantum effects, and we showed they went away with anesthesia. In the meantime, a lot of other work, Honorbaum's work and work by Travis Craddock has shown a correlation of anesthetic effects on microtubules that correlate with their potency in putting people and animals to sleep. So I claim, and I think it's true, I haven't heard anybody refute me, that our theory, ORCO-R, has far more explanatory power, and I can go through everything, all that. Connection to biology, because they don't have any connection other than cartoon neuron, and experimental evidence. And they don't have anything. But they just continue to ignore us. And I don't get it. But anyway, it'll take some time. I'm getting old. I hope I'll last long enough to see it.
Yeah. So what happens? So all right, microtubules within the context of Alzheimer's disease dissociate, tau falls off, you lose cognition, as you mentioned earlier, right? But with aging, does that happen independent of Alzheimer's disease, where the microtubules will dissociate without having Alzheimer's disease?
I think so, probably. I just Googled something in preparation here, and I found a paper about well, I don't want to go find it, I'll lose you, but about microtubules and longevity. And it was in animals, and they looked at the state of the microtubules as a function of the longevity of the animal, or the other way around. And they found a good correlation. So I didn't get a chance to study it, but I'll look it over and I'll send it to you.
Yeah, that would be great. Then the other side is when considering that microtubules are linked with cognition, and I think I saw in one of your presentations, you posted a slide of the density of microtubules in the brain based on species, right? With humans.
Oh, yeah. The number, the total number, basically based on I getting the information here and there. Yeah. And, so in our theory, conscious events occur at time t equals h over E sub g. So h is Planck's constant. E sub g is the gravitational self-energy of superposition, of pulling something apart from itself. So with the sun, well, the sun's not in superposition, but one atom nucleus would be a fairly small amount. That's E sub g. So that would take a very, very long time to reach collapse. So one atom take 1 million years, something like that. For something large, it would happen very quickly. So biology, you want it to happen at the range of neurophysiological effects. So in our original work back in the mid-90s, we thought, well, that must be like gamma synchrony, 25 milliseconds. So we said t equal 25 milliseconds. We soon realized that's a long time to avoid decoherence. And number two, that's not very many microtubules. It's very few, actually, almost too few. So, but we thought, well, it could happen faster, and more likely to happen faster, like at 10 megahertz, which is a resonant frequency for microtubules. And if it happens at 10 megahertz, then you would need a lot more tubulins in superposition, 10 to the 15th. So, and they would need to be entangled. And that seemed like a big ask. But now Honorbaum has shown entanglement between microtubules, and in the nonpolar regions, I don't see why they couldn't be if they're oscillating coherently. But the other problem was that, our cognition and consciousness, it doesn't happen that fast, 10 megahertz. Most people would say we have epochs, like visual scenes, gestalts, roughly 100 milliseconds. So how do you get from very fast 10 megahertz events to fairly slow cognitive epochs. And Roger and I revised our paper in 2014, and we talked about this. And we had talked about interference patterns. And he said, well, it could be negative resonance or interference from the very fast going down and down. And that's exactly what Anirban has shown. These time crystal behaviors with repeating patterns every 3 orders of magnitude. So we said, okay, well, there's interference, and that's how you get down to it's kind of a downshifting. And actually, the work of Earl Miller at MIT and Dimitris Panatsis and others has been showing this downshifting is very important. So in feed forward from the back of the brain to the front of the brain, you go from very fast to very slow. And in the cortex, you go from the top of the cortex, layer 1 and 2, down to layer 5, where the pyramidal cell bodies are. And that also goes from fast to slow. It's in gamma synchrony up at the top at the apical dendrites, but as you go down towards the pyramidal cell body, you get down into alpha. So you're going from roughly 40 down to 10. And I think that's also an interference pattern. And it's kind of like you're continuously or repeatedly focusing in on something. There are each little collapses, and you're getting rid of more and more unnecessary stuff, and eventually you get to the solution, the conscious moment, the decision that you want to make. You collapse on that, and that's your reality. And then it happens again and again and again. So that's what I think is going on.
But that's faster than millisecond timescales.
That is, but then you get to the epoch. So if you get to so when you get to the 10 or 100 milliseconds, you're down to a very few number of tubulins, but that's all you need at that point, I think. If you got you got rid of all the unnecessary stuff.
All right, wait. So where I was going with the slide with the microtubule density for based on species with humans at the top, and I think at the far right you had like maximum human computational ability, right? So that implies that there's going to be a range that people have to maximize their cognition, potentially by, is it possible to maximize microtubule density and to resist any age-related decline? Has anyone even looked at that? Or is that even a thing?
Well, you do want to keep your microtubules healthy. That would be the main thing.
Yeah.
I don't know if you want to increase your density. I think at this point, you want to keep the ones you got healthy. And, we've seen these pictures of aliens with big heads. So maybe they got, 10 times as many microtubules, who knows, but we got to get there first. So, the main thing I think is to keep your microtubules healthy. And one way to do that, if you have a problem, is with ultrasound, as I was talking about before. But I'm sure all the vitamins and nutrients and everything else that healthy people take probably do that also.
Well, plasma,
pardon me?
I was going to say plasma levels of tryptophan decline during aging. So that's probably not contributing. Yeah. That's at the core, right? So
yeah, we want more tryptophan. And tryptophan is the aromatic amino acid with the indole ring, which has the highest energy, highest excitation energy in fluorescence. So that's why microtubules absorb UV a lot. And there's probably UV in the cell for mitochondria. And so you want to take care of your mitochondria too. A lot of people think it's all about the mitochondria, and maybe to some extent, at least it is, but they produce UV photons, which are very readily absorbed. So for another added advantage is you're getting rid of potentially toxic metabolites or toxic radicals that way. If you, it does two things. The UV, I think, fuels the quantum processing of the microtubules, and the microtubules, by soaking them up, get rid of any negative effects.
All right. So you mentioned the ultrasound as a potential Alzheimer's therapy, but on the other side of it, so ultrasound is what, 20,000 hertz? Could you, is it possible to have like, I don't know if there's...
It's more like megahertz, actually. Well, anything above audible, which I think is 20,000 hertz, up to several millions of hertz is ultrasound. So when Anirban discovered that microtubules had coherent oscillations in these different frequencies, kilohertz, megahertz, gigahertz, terahertz, this is back in about 2010. Well, he published on it. I met him in 2009, so it was around there. I started to think maybe we could treat the microtubules with oscillations with one of these frequencies. So kilohertz, that wasn't interesting. Megahertz in electromagnetics is radio waves, and I wasn't interested in putting radio waves into anybody's brain. Gigahertz is microwaves. I wasn't interested in that. Terahertz is photons, and that's pretty hard to get in, although people are trying to do that. But if you go back to megahertz, I looked over and there's an ultrasound machine, which we use in anesthesia all the time, and to image, looking for the jugular vein or do nerve blocks or whatever. And I'm thinking, I wonder if anybody's tried megahertz ultrasound into the brain for mental effects. So I looked it up, and there was a guy at ASU just up the road from us, Jamie Tyler, who had been looking at ultrasound into rats and putting electrodes in them and looking at behavioral effect. So, he could zap the right motor cortex and make this paw move and stuff like that, or cause physiological effects. Of course, you couldn't tell how the rat was feeling with it. But I also found out that it was approved for brain imaging before, CT and MRI made it obsolete for that, and was still used to look at newborns for bleeds through the fontanelle where there's no skull at all. And so I figured, well, it can't be that harmful. We put it everywhere. Why not the brain? And it doesn't seem to be a problem. So, and we had a pain clinic, and with a lot of the patients are depressed, as you might imagine. I used to run the pain clinic, and I knew that. And I said, why don't we try this on our chronic pain patients who are depressed and see if they get better? Maybe their pain will get better. Maybe their depression will get better. And they're kind of laughing at me. Yeah, right, Hammer. Well, I'll tell you what, your idea, you got a shaved head. Go right ahead. We'll watch. And so they called my bluff. And so the end of it, a long day in the OR, we're sitting around the table, and I said, okay, here we go. So you put this goop on, and I held it up to my head. I didn't know where else to put it. I knew that this was a thin area of the brain. And I was kind of watching myself on the screen. You can actually see the temporal pole and the cortex. And I held it for about 15, 20 seconds. And I didn't feel anything. So I was really disappointed. I put it down. I said, oh, well. But about a minute later, I started to get a buzz, and I was kind of high for an hour. I mean, it was the end of the day. I was going home, fortunately, but I felt really great. And I said, we got to do the study. And we did, and published in early 2013. It was the first study on ultrasound effects on human mental states, or mental states at all. It was published in the journal Brain Stimulation, a good journal. And so a lot of people started doing ultrasound, but ultrasound can be focused. And if you use TDCS or TMS, you can't focus it. Ultrasound, you can focus like a laser beam on any part of the brain. And so people go and say, oh, we can stimulate the hippocampus, so we can stimulate this, stimulate that. So they went off in that direction. And I thought, well, why are you bothering? Because it's going to spread out anyway, and you're affecting microtubules. So people, so most of the work since then has been done in focused, and also high intent. If you do it at really high intensity focused, you can zap a lesion. So if you got a EEG, a seizure focus, or a tumor, or a pain focus, maybe you can zap and get rid of it by burning the brain. I don't want to do that. At a medium doses, you can open the blood-brain barrier to get drugs across that wouldn't get in. I don't want to do that either. Low intensity, without heating at all, you don't do anything except, who knows, resonate microtubules. Although people in ultrasound don't want to believe that. They think it's activating stretch receptors. I'm going, well, how does that do anything? So anyway, it's controversial. There's a fair amount of lab stuff showing microtubule effects directly on microtubules, but it's still controversial. Most people will say it's stretch receptors, but I don't believe so.
So I was thinking the opposite side of that equation in terms of, so not excitation and excitation frequency. Is it possible to have sound-induced anesthesia, right? Maybe you generate an interference pattern that basically damps down the microtubules just like chemical anesthesia would. Is that even possible?
It might be. My friend Jay Sanguinetti, who started doing a lot of focused ultrasound, was looking. He said, well, if you ultrasound here, it's inhibitory. If you ultrasound here, it's excitatory. And I never quite believed that. Anyway, we have good anesthesia. We don't need ultrasound for anesthesia. What we don't have is a treatment for Alzheimer's. So whether it can be people are using TMS for anesthesia. I think Tononi patented. I'm going, why? We got really good anesthetics. And I'm not going to put that damn thing on my patient's head and have to worry about that crap. Why? We have really good anesthesia. So I don't think we need better anesthetics. Propofol is great. Sevoflurane is great. The drugs are great. we don't know how they work, and we don't know how to fix the brain when it's broken. That's what I'm more interested in.
Yeah, gotcha. All right. So when considering the quantum effects of microtubules, so when I hear quantum effects, I start thinking about entanglement. Is that entanglement local to the brain, or now are we talking about entanglement of processes happening in the brain that are linked to somewhere else? Who knows where else? So what do you have to say?
Yeah, good question. First of all, I think we need entanglement in the brain just for things like cognitive binding, spatiotemporal binding. So visual images start on the, V1 of the back of the brain, move forward, and they go through associative cortex, and you add shape, color, motion, meaning. And when it gets here, you got the whole picture. It's a blue bird, or it's a red kite, or it's a silver airplane. It's not, something flying with silver and this and that. So it all binds together, despite those different features being processed at different places and different times in the brain. We don't see different time. We see one thing. So there's spatiotemporal binding. So not just spatially, but temporal, which entanglement can do. So I think we need that just for normal brain function, and we need it for zero phase lag gamma synchrony, EEG, at the high frequencies where it's perfectly synchronized over the whole brain. That's really hard to explain by traveling membrane potentials, and even ephaptic electromagnetics, because that drops off with distance very rapidly. So I think we may need entanglement just for normal spatial temporal binding, and definitely for ORCA-OR, for all those tubulins to be entangled into one, so that you have one collapse at every 10 at 10 megahertz. So, but you're asking a different question, and so we have this image of a hierarchy going from the neuron in hertz to faster, 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 faster. We get to where anesthesia works in the terahertz and petahertz, and we keep going faster, and eventually we get to the Planck scale. It's a big step. The last step is, because it's 43 hertz, 10 to the minus 33 centimeters. But, Roger's idea would suggest that these processes, quantum processes happening in the microtubules, go all the way to the Planck scale, and that where the qualia. And the Planck scale is in here. It's everywhere. You don't have to go for it. It's right there. It's just at a much, much, much smaller scale. But once you get faster than, 10 to the 18th hertz, you're smaller than matter and you're faster than biology. So it's probably non-local there. And so, like in near-death experiences, when the brain stops getting oxygen and blood stops flowing, eventually at some point the quantum information in the microtubules kind of dissipate to the universe, but could stay entangled as kind of a soul. So you could have an out-of-body experience, a near-death experience, out-of-body experience. The patient is revived and they go back inside and they say, "Oh, it's floating above," or they're not revived and we don't know. But there's a lot of pretty good evidence for reincarnation, actually, when you hear these interviews of these kids who have memories, and then they have birthmarks that correlate with the way the person died and this and that. So until we know that consciousness in the brain is strictly local and classical, we cannot rule out non-local consciousness outside of the brain.
How would you even test?
I'm now thinking that consciousness preceded life. I'm now working in astrobiology with my friend Dante Loretta, who was in charge of this NASA mission to an asteroid to bring back organic molecules. And he's been interested in the origin of life his whole career. And he was assistant to this director on this big project. Dante was but the guy Mike Drake, unfortunately got sick and passed away. And Dante at a very young age took over, and it went great. And the samples came back last September. He was there. I watched it on TV. And so we're analyzing the samples now. So anyway, while he was waiting, he told me he had promised, to unveil the origin of life. So how's he going to do that based on these molecules? And he got the idea that evolution would make a lot more sense in terms of the behavior of primitive creatures, just moving around doing stuff, if they had some motivation. And there were no genes. So maybe they were conscious in a primitive way, right from the start, right from the origin of life. Feelings. Something feels good, so I'm going to do that. why am I going to go here? It feels good. Why am I not going to go there? It feels bad. Simple stuff like that. And he said, I wonder if anybody else in the world has had this crazy idea. And he went to Google and found me. And I was right across the street. And we didn't know each other. And he said, who the heck is this guy? So he emailed me and I said, well, that sounds interesting. So we met and he told me his idea. And he saw a paper I wrote called The Quantum Origin of Life. Did the brain evolve to feel good? Which basically suggests that Rogers' objective reduction, which gives qualia, gives feelings, occurred in the primordial soup, which had these aromatic rings. We know that from the simulation of the primordial soup. And so I suggest in this paper that there started to be conscious moments in the primordial soup with these molecules, micelles, they're called, and or arrays of molecules. And some of them, at least occasionally, would feel good, would be pleasurable. And that would provide a feedback fitness function for these micelles to proliferate, to develop, to self-organize, to optimize to optimize pleasure. And that was the origin of life. That's what I suggested. And even evolution. Evolution's a lot easier to figure out if there's pleasure. Why would anybody do anything for their genes if their gene, the Dawkins's idea, gene survival. If genes don't have feelings, why would that matter? It doesn't make any sense. It's it's teleological and so I think pleasure from the start is probably the most logical thing, which means consciousness was there before life. And how long before life? Well, who knows?
Well, in the self-assembling molecules that had aromatic rings that could I basically capture quantum moments, or maybe yeah, I don't know how that would, you know what I'm saying? So
well, yeah, so this is also interesting because these aromatic rings are floating around the universe. Stars, young stars produce aromatic hydrocarbons, not just benzene like goes in our gas tank, but complexes like little bits of graphene. We have ring complexes in planes, but also buckyballs and fullerenes. And the carbon nanotube fullerenes, where you have a cylinder of aromatic rings that geometrically looks a lot like a microtubule. And we're I can't say too much about it, but we have reason to believe that suspect that microtubules were present way earlier than evolution would have indicated. And that's all I can say at this point, because it's almost too crazy, but we do have some evidence. We're going to follow up on it. So, which means that maybe the only way that could be, I think, is that these fullerene nanotubes might have themselves started having conscious moments and kind of beefed up and became biological in some way at some point. And they were the origin that these nanotubes became microtubules as the starting life, kicking off life.
So when I've heard you talk about consciousness existing outside of life, I mean, that suggests that it's not bound to any structure, right? So that is what you're saying, or that's not what you're saying?
It could be in space-time geometry, right? I mean, which doesn't have structure. It's space-time. I mean, it puts in the realm of general relativity rather than classical physics or quantum mechanics. And it's hard to measure that kind of stuff, as it is hard to measure consciousness. I think the only way to do it, and I don't know if this is possible, but in principle, the way they measure gravity waves, with these mile-long LIGOs in perpendicular, and which look a lot like centrioles, which are made of microtubules, but that'd be probably too much to hope for. But so yeah, I think it's more in the realm of gravitational waves that are so tiny that only quantum, aromatic rings or electrons can detect them. I don't know, but yes, they can happen in space-time geometry without any matter, due to space-time curvature, and yeah, and plus there is some matter floating around there even in the vacuum. But yeah, you can. The idea is purely space-time geometry. It could have separations and collapses.
So how would we know if we're tapped into, if there is a consciousness that exists outside of brains? How would we know that we were tapped into that and we weren't just staying local within what's inside of our own?
Well, that's a good question. I kind of think that when we go into meditative states or altered states, or just feeling good or intuitive that we tap into platonic information embedded in the universe. And Roger brought up these platonic values in his books, and then I probably pushed him too much, and he's kind of backed away because it sounded too religious or too spiritual, which he tends to avoid. But I think that these platonic values, they influence the collapse, which is not which is not random nor algorithmic, but influenced by these platonic values. Could be something like, the way of the Tao or divine guidance or something like that. It's not religious. I think it's just physics, but
Then
physics is pretty spiritual.
yeah, maybe. So are the quantum effects, are the frequencies that are in microtubules, are they different? Like, for example, during sleep, during the different stages of sleep versus when being awake? Like, is there a differential tuning of microtubules on differential states?
Well, we don't really know yet, but we hope to know soon. We know that if you just look at EEG under anesthesia, everything slows down. The gamma goes away and everything kind of slows down. We're thinking that, and Anirban just today published a paper on megahertz and gigahertz EEG, and we're planning studies this summer. It's going through the ethics committee now at 2 hospitals in Bangalore in India, where his colleagues are going to try this megahertz on patients going under anesthesia, and we'll see if it goes away. I'm hoping that the megahertz and the gigahertz will be far more sensitive than EEG. I think they will be, and then it could be a monitor for depth of anesthesia. Not that we necessarily need that, but it would. They also want to try it in comatose patients and see if there's any signs of, and see what that shows. So there's all kinds of possibilities. So this is just brand new stuff, and I'm hoping that mainstream labs will start doing it. I'm talking to a few, but I think that'll happen. And people are going to have to do it just to rule it out, to show we're crazy. And I don't think they're going to be able to do that.
All right. So do microbes have microtubules, or do they have structures similar to microtubules? Like, for example, gut bacteria or skin bacteria, do they have microtubules where?
So yeah, good. All animal cells and all plant cells have microtubules,
But not
prokaryotes and Archaebacterium have a different protein polymer that is very similar, but doesn't have tryptophan. It just has phenylalanine and I think tyrosine.
Yeah.
So yes, they can have conscious moments, but they would be kind of dumb compared to ours, because they don't have the energy of the tryptophan. They're starting from phenylalanine.
I see. So then there's this balance, right? Because we're probably about half microbial and half human genetics. There's this balance between the dumb consciousness in microbes in our gut, skin, everywhere else, versus the tryptophan focused, consciousness in our.
I don't know about that. I know the gut bacteria, and people think that they're and maybe they are, but I don't know that they're entangled. I don't know if they're entangled with each other or with us. And so I worry about the consciousness in the brain. It certainly can be elsewhere. I know when I used to do cardiac anesthesia and we did heart transplants, that the surgeons would tell about patients who had never danced a day in their life. They got a heart from somebody who was a big dancer. Now they're dancing. So maybe there's memory in the ganglia in the heart. I don't know. I mean, that's not a scientific proof of anything, but anecdotal. But a lot of that stuff does happen. And I think memory is encoded in microtubules. Where else could it be? Because synaptic proteins last hours to days, and memories can last lifetimes. So it can't be in the synaptic proteins. And the microtubules and dendrites in particular are perfect, because microtubules and all other cells, when it's time to divide, they have to disassemble. So imagine a microtubule lattice is encoding information, kind of like a mosaic. And then, okay, time to divide, it disassembles, they all kind of jumble up and then reassemble, you've lost it. Neurons don't divide. So once those dendrites, and then they get capped on both ends, so they don't treadmill. They don't lose from one and add from the other, or what's the other thing? Dynamic instability. They're quite stable. And each tubulin, so there's about 1 billion tubulins per neuron, and each tubulin can have at least 30 different states: genetic, post-translational, phosphorylation state. And we've shown how memory can be coded through an enzyme called CAMK2 from synaptic calcium coming in and coding on the microtubules. So there's a billion to the 30th power of possible memory states in one neuron. So multiply that times 10 to the 11th or whatever. But I think, we want a lot of redundancy. We know memory is distributed. It's not in any one place. So we want it to be repeated over and over again. And it's probably in the microtubules all over the brain. So we got some resilience there, but eventually, when the microtubules fall apart, you lose memory.
Gotcha. All right. Some random questions. Is that hopefully okay?
Yeah.
So I think in most, I don't think I've ever seen you ask these questions. So, and they're going to be totally random, but like, what's fun for you outside of studying consciousness and microtubules and astrobiology? Like, what do you have favorite movies? Can you even watch movies? Because most of them are just too, banal and boring. And, so what?
No, I love movies and I love sports.
Yep.
I'm a huge basketball fan, University of Arizona Wildcats. I have great tickets. I've had seats since well before Lute Olson, if you know, ever
Yep.
heard of him.
Yeah.
In the 70s, I had season tickets, and I'm in the second row behind the visitor bench. I'm there almost every home game. You can look behind the visitor bench and see me. And I love my dad was kind of a sports addict and a gambler. Unfortunately, I didn't get the gambling bug, but I got the sports addiction. So if there's a game on, I'll have it on. I may have it off, the sound off. I turned it off when we started because I didn't want to be distracted. I'm watching the Euro Cup and the golf and whatever's on. I love sports, and I used to be pretty athletic. I've got pretty severe arthritis. All I can do now is hiking and pickleball. I might ski. My legs are okay. My right shoulder's pretty bad. But I'm in decent shape, and I used to be pretty athletic, and I was kind of guy who was decent at everything, not great at anything. Baseball was probably my best sport, but I still love sports, and I love culture. I love people. I love everything. I love hiking around here. I live in a beautiful area. I'm looking up at the mountains here, and it's too hot today to go hiking. But as soon as we're done, I'm going to jump in the pool. And my son and his family are coming over for Father's Day, and he's cooking, and I'm looking forward to that. And so I hang out with their kids and watch sports and just relax.
So all sports, what about like MMA or UFC? Do you follow that stuff too?
A little bit. I don't know. It's a little too violent for me. I love Muhammad Ali. I saw when I was a kid. I can't remember exactly when, but he was my hero.
But from the boxing point of view, from the
Both.
Yeah.
I met him once in Philadelphia. He used to just hang out in the street corner, and yeah, I just liked his vibe.
Nice. All right. So favorite movies? Do you have
Ooh.
genre, even genre that you
I
like?
kind of like Humphrey Bogart movies. I kind of relate to him for some reason. Uh, um, I like action thrillers. I like, movies like Contact or Deep Impact and even Armageddon, although it's kind of corny. So I like anything, really.
But nothing stands out where it's like, this is my movie. I'll watch it.
Probably my favorite movie is The Big Lebowski. Uh, and I watch The Godfather anytime it's on. I learn a lot from that movie. Not that I want to be like them, but how other people are, and I recognize it when I'm getting muscled. And so I watch that, and I watch a lot of. I'm looking forward to House of Dragons coming back today. You know that show?
Yeah, I'm not. No, I know of it, but I don't watch it.
Okay, so I like that. So I like stuff like that.
All right. So also, I think you
And
retired.
I like travel. Excuse me. I love to travel. I just and one of the things about this whole deal has been it's gotten me to see the world. And when I first started in anesthesia, my future chairman said, and it's a lot of fun. And he was an academic. He introduced the anesthetic sevoflurane into the American market and figured out halothane hepatotoxicity. So after I was in the department for years, he kind of disappeared, started traveling all over the world. And I realized you could do that. And so I've gotten to see the world a lot just through the research. So I love to travel, love to go places. I just got back from the How the Light Gets In festival in Wales, which was really a blast. And I'm going to another festival in July in Barcelona, the Festival of Consciousness. And we'll be doing our conference, The Science of Consciousness, as a lead up to their festival next year. So Barcelona has always been one of my favorite cities. I'm going to take my son, and he wants to go deep sea fishing, and I want to just sit on the beach, and we're going to have a good time.
Nice. All right. So you were in academia for 49 years. I think you retired earlier this year or going
Well,
to.
I retired from anesthesia, but I'm still I'm still at the University of Arizona, and I've changed colleges, but I'm in astrobiology, and we may change it to astrobiology and astroconsciousness. We're thinking about that.
Nice.
We got to
So
find it first.
got a what first?
We got to find it first.
Oh, right,
We got
right,
to
right.
find signs of consciousness in these molecules.
Yeah,
And I'm writing today. We're working on a proposal how we're going to do that.
nice. Looking
Basically,
forward.
if we get collective oscillations, kind of like microtubules in these molecules, then we're going to see if they go away with anesthesia. And if they go away with anesthesia proportional to the potency, the known potency of the different anesthetics, and putting you or I or any animal to sleep, then we'll claim that this could be the roots of consciousness, and maybe consciousness was there at the beginning of life.
Their quantum effects would have to be a part of that story, right?
Yeah, that's what we're looking at, quantum effects.
Right. So if you've got molecules and you've got quantum effects and you've got potentially consciousness, that goes back to where is that entangled particle? Where is the particle that it's entangled with if it's an isolated molecule and not a giant body, right? Or a brain.
Well, one particle, yeah, one particle would take a long time to collapse. So you need like a micelle of a complex, and some of these micelles get pretty big, and you get a lot of aromatic rings. So you need a lot of them, and they would have to be unperturbed for a period to have the conscious moment. Or they could be floating in space, where they're in ice, where exposed to UV, and it happens that way. I'm not sure. There's a lot of possibilities, and I think nature found a way somehow.
All right. And then advice for young scientists, like what would you offer up? That's one. And then two is if you could go back in your career, are there things you would change? Now, granted, the things, I wouldn't say the things that have led you to where you are now, but the things that you would change, basically offering other young scientists advice and then young advice in your own career. You know, those two, how would you address that?
Well, as far as advice to young scientists, particularly neuroscientists, I'm really worried that I mentioned the two groups in consciousness studies, the ASSC, which is following the cartoon neurons, which is heading into a big suck up to AI, versus our group, which is more open-minded. And I think AI is funding a lot of research now in the cartoon neuron labs, because all of a sudden they're advertising, oh, we have postdocs available, we have this and that available, come work with us, blah blah blah. And this is only, I think, only happening to the it's happening at the cartoon. So I think they're going to suck a lot of young scientists into dead ends with these cartoon neurons. For example, Christoph Koch and Dave Chalmers and Giulio Tononi just promoted the hell out of IIT, which I thought was the stupidest theory I've ever. It's pure information. It could be anything. A thermostat could be. There's no biology at all. And then when there was this letter from 125 neuroscientists saying that it was pseudoscience. I said, well, I kind of have to agree, And then Dave and Chalmers and Tononi and Christoph got all, oh, you're ruining the careers of all these students who've been working on blah, blah, blah. I'm going, no, you ruined their career by touting a bunch of bullshit that they spent years working on. Don't blame me. So I think that's happening on steroids now. If the cartoon neuron people get sucked up into AI, and it's all going to be about brains equals computers, and forget about all that other stuff.
String theory, I guess, has had some of that same problem, right? For the past 50 years or so, I see a
String
lot.
theory, yeah. Well, even I don't know how old you are, but before that, it was remember the superconducting super collider? All the physics people went into that. They was going to build this big collider in Texas, $1 trillion, and then they canceled it. So I don't know what happened to those people. I guess they went into string theory, and now they're unemployed, or they went into chaos and emergence theory, and now they're unemployed. I think all these things are fruitless.
Yeah. So the masses usually follow the wrong approach and don't know that they're following the wrong approach, right? It may be a limitation of the game.
I think they're following the wrong approach, and I think they're going into a dead end, and they should wise up.
All right. So what advice would you give to your younger self?
I've sometimes wondered. I got an offer to do research when I was still in medical school at Penn in a very prominent biophysics lab by Britton Chance. And I was at the end of medical school. I wanted to move out west. I didn't really want to do what he was doing, which was mitochondria. And then I thought I could have done a PhD at other times, but I don't think any PhD I could have done would have taken me where I am because, they were all following conventional. If you do that, you got to do NIH and you got to write what NIH wants to hear. You want a project that fits within their guidelines, not too risky. And so I think we're really losing our innovation, competitive edge, and I think AI, knuckling under to AI and conventional this and that. So I'm not sure I would change it. It's been a rocky road in a lot of ways, but I've had a good life. I can't complain.
Nice. It's a tricky road, right? Because if you've got a great idea with some preliminary data, you may get a group of scientists who review it who aren't outside the box thinkers and don't see the beauty of it because they're conventional thinkers, and then it doesn't get funded. And then what do you do then, right? So I'm actually thinking about leaving academia. I've been in this for 2003. I got my PhD. I'm just tired of reviewers reading 3 paragraphs, and I'd like to think I write well. 3 paragraphs of my grant, and then saying we're not interested in how gut can impact muscle, the gut muscle axis. And meanwhile, 10 years later, there is a gut muscle axis, right? So, but I had to deal with the reviewers who, again, it's not as groundbreaking as the stuff you guys are doing with consciousness, but it's still within my own little niche. So yeah, so I'm thinking about leaving academia. I'm tired of dealing with reviewers that lack.
Or find the right spot. I mean, I lucked out tremendously because I wasn't sure what I was going to do when I finished anesthesia. I knew I wanted to keep going in consciousness, and I ran into Dante, and it really fits. So I lucked out there, and he's become a really good friend, and he's a very smart guy and very capable. He's much better at organizing and grant writing and administration than I am, and that was a big weakness of mine. So I never ran a big lab, or I was good in the operating room, and I was good in thinking and theorizing, but I wasn't good in organizing too much. Although I organized the Tucson conferences, which have turned out to be really great. But
nice. All right. So is there anything we didn't cover that we should have covered? What does ORC-OR get wrong? Is there anything that it gets wrong? Is there a flaw that needs to be addressed that hasn't been addressed yet?
I don't know. We don't know what space-time actually is. Roger's the world's expert, and he doesn't know. It's twisters or spin networks or quantum gravity or loops, or we don't really know. But and some people think it's not fundamental, like Don Hoffman saying, well, you got to go deeper and deeper. Well, maybe you do, but getting to the Planck scale will be enough for me at this point. The other question is, Rogers also went to what was there, the origin of the universe. He thinks the Big Bang was preceded by another eon, which had another eon, and we've had a series of eons going all the way back. And it wasn't a Big Bang; it was more like a big brush fire. And this eon will end, and then there'll be another one. And so, was there consciousness in the previous eon? Who knows? I mean, it's kind of like now that consciousness is embedded in the fine scale structure of the universe in space-time geometry. That was one of Rogers' brilliant ideas. And if so, maybe when the eon ends and there's a new eon, that it's kind of like a mutation and evolution, that the cosmological constant, the 22 numbers that make up the universe, might evolve and mutate a little bit, so the next eon will be more suitable for consciousness, to optimize consciousness. I mean, what else would be the purpose of the universe other than to optimize consciousness? I don't know. That's just something that we've talked about. We're kind of far enough out on enough limbs that we don't have to talk about that now. But it's interesting that the consciousness may be eternal. And somebody at the Scott Aronson, the quantum computing guy.
Yep.
Brilliant guy. We've kind of tangled a few times. And I said that at the how the light gets in festival, and he goes, "Oh, so we're gonna have this loop." Oh no, he said, well, what if you go all the way back, all the way back, all the way back? When did it start? And I wasn't sure I should have said this, but I said, well, maybe if you go all the way far enough back in the past, you wind up in the future. It was kind of a joke, but he took it seriously. Okay, so you have this big loop. How are you going to prove that loop? I'm going, I don't know, Scott. Let's talk about it over beer. So who knows? What started it all?
Good stuff. Yeah, I don't, who knows, right? Who knows if we'll ever know, right? That actually gets back to the reincarnation issue. So if that is possible, then why doesn't everybody have a reincarnation experience? Why is it in this very small, literally very small minority of people of the billions that are on Earth, right? How do you reconcile that?
How do we know everybody doesn't?
Wouldn't we have that experience in some way? I mean, would you have to?
People who have good stories forget them as they grow older. So maybe we forgot them in the uterus, or maybe it wasn't traumatic. Maybe you only remember traumatic things.
So could cryonics be a death sentence in terms of frozen forever, not allowing, if there is a quote unquote quantum soul, I think as you put it, is that like frozen in carbonite like Han Solo, and you're not allowing whatever that quantum is to leave?
Well, microtubules don't like being cold. They depolymerize. So we had a session on that, the 2014 conference on the future of consciousness, and we had this cryo guy. And basically he said, well, we have to choose between preserving the membranes and preserving the microtubules. And so cold is the way to preserve the membrane. I said, okay, well, there goes consciousness. Forget it. Well, he didn't like that very much, but I haven't heard much about cryogenics since. And I certainly wouldn't do it.
Yeah. So that doesn't leave many options for those of us in the longevity community who have no interest in death, and cryonics may be out and don't want to be buried in the ground to be worm food, right? So I guess it's like
I used to worry about that too, and now I really think that there is quite a good likelihood for consciousness after death, and if it's really in space time, so I've stopped worrying about it.
So that's almost brings us into the psychedelics realm. So DMT, right? So are those realms somehow a different? Are microtubules somehow different under psychedelics? Whether the frequency that
we want to study that, and my friend Travis Craddock has looked at psychedelics binding to microtubules and find that they do, and we think that they increase the frequency of the oscillations. So if you go back to that hierarchy, I think that would. So anesthesia makes it slower, and psychedelics would make it faster. So there's more conscious moments per second. So if you think of that other graph, we'd move into we'd move from maybe 10 million conscious moments a second up to whatever the max was. I forget the total, 10 to the 20th tubulins in the brain. So there is a lot of room to go higher, faster, more conscious.
So then, I mean, it's almost like we're tuned into a different frequency of the universe then, potentially.
We go to a higher frequency, yes.
Yeah. Huh. So could that be a real realm? I mean, people under DMT experience to see all kinds of crazy stuff, elves and all kinds of other, so what are your thoughts about that?
Could be archetypal images embedded at some level of the universe, Jungian archetypes, platonic values, something like that.
Interesting.
I think, in that experience, they're real. I mean, they seem as real as you seem real to me right now, and they probably are.
Cool. All right. Well, I think we covered it all. Happy Father's Day. Enjoy the day with your family, and thanks, Stuart. Ciao.
Thank you, Michael. Good meeting you. Take care. Bye-bye.